everyone. My name is Kristianna Sarkin.
And my name is Lauren Urban, and we're both members of Reach Out Teach Out at UCI. Today we're going to be talking about a fundamental part of scientific studies that comes after the experiment is done and you've collected your data. That's statistical testing. In this video, we'll focus on the importance of statistics and how to know when to use the proper test. We won't be focusing on the mathematical equations that are used because there are many modern programs such as Microsoft Excel, Google Sheets, graphing calculators, and even R that can help you with these parts of the calculations. For more information on these tools, please refer to our Roto tutorials on data management and analysis. To compare two groups or treatments, we need to know the average for each group and its variability. This is known as the mean and standard deviation. We can see here that both the average weight or the mean and the standard deviation for each group are shown below. But let's visualize it in a different way. Let's say that we can consider the average weight to be the bullseye in the center. If we look at group A, we can see all of the dogs are very close to that average weight, varying between 70 to 80 pounds. Let's compare the second group. As you can see, there's a lot more variability, meaning distance between where the points are located in the second group. This makes sense because the variability or standard deviation we saw was much higher in the second group. So, you might be wondering, how does this relate to scientific studies? Well, scientists often have hundreds of data points for a single experiment. As a result, it's often more effective to analyze trends in data instead of summarizing all of the raw data points. It's equally important to make sure that we use statistics in order to analyze differences that we see in our data and determine whether they are real or not. As we can see in this example, we have two different bar plots, each showing how different age groups perform in terms of their mile time. If we saw this group in orange, we might think that there is a statistically significant difference in terms of mile time for each of these different groups. That's based off of the mean, which is the height of each of these bars, as well as the small error bars, which indicate a small standard deviation within that group. In comparison, the group in blue shows a lot of overlap in terms of the mean values. We can also see that the variability or the error bars are much higher in this bar plot. So we might not believe that there is a statistically significant difference in terms of mile time between these different age groups. Although it's important to note that you can't just tell from looking at a graph whether or not there's a statistical significance. You always need to perform the actual test to confirm. But these plots can give us a clue as to whether or not there are trends that we can test later on. So, what type of statistical test should we use? Don't worry, we're not going to go over all of these examples, but this is just here to show you that there's quite a lot of different tests that are very specific depending on the type of data that you have, as well as your experimental design and the distribution of your data. We won't go into too much detail about most of these, but we did want to provide this as a reference, as well as a couple of links in the YouTube video tutorial below. But before we start talking about the correct statistical test, let's understand two important concepts. The first is different types of data. So there are different measurements that we can collect in an experiment. But in terms of data, there are two major types that you'll be working with. Either categorical, which is going to be anything that's a non-numeric output that you're looking at. This could be gender, favorite color, eye color, things like that. The other type of data is numerical. This could be either discrete, such as a very specific number, like the number of siblings. You won't have 1.2 siblings. The alternative to discrete is continuous. So there are discrete numbers in between the intervals that you're looking at. So for height, you could be 5.2, 5.3, and such on. The other important thing to know is that a lot of statistical tests are based on the normal distribution. This is because a lot of nature actually falls under the normal distribution. If you're looking at heart rate or weight in general, most of the time it will fall in a normal distribution for a population. This means that a lot of statistical tests and the analysis that they're based on rely on this bell-shaped normal curve. In order to make sure your data is normal, you can use a plot, such as a bar plot or a histogram, to see whether or not it is skewed to the left or to the right. We won't go into too much detail about that, but here's a great link with a tutorial about how to tell if your data is skewed or not. You can also perform statistical tests to tell whether or not the data that you have is normally distributed. So, when it comes to picking the right statistical test, it's important to understand the type of data you're collecting, your research question, and the way you designed your experiment. This will all inform the best statistical test for your data. The three most commonly used statistical tests in biology are the t-test, ANOVA, and chi-squared analysis. We'll go into detail and provide an example for each one so you can get a better understanding of when it's appropriate to use them.